Good morning once again. I'm Dixon Rotojera for Ifakara Health Institute, and I would like to thank Africa Genetic Biocontrol Consortium for this opportunity. Professor Tony has been kind enough always to invite us in this platform. And I think apart from developing the technology, when we hear science communication, we always take this serious because it's an opportunity for us to learn. So I'll give an overview of genetic or as engineering biology project that we're working at the Institute. Aside from what Dr. Kayonda has pointed out on different things happening at UVLI, at Ifakara, this is our flagship program. And we are using it to build different platforms where you can also welcome different engineering biology projects that we might be able to work on in the future. I think we all know why we are working on genetic engineering for addressing malaria problem. So much has happened in the public domain in terms of controlling malaria. But even when you look at WHO 2023 report, you can see that we're still encountering over 600K deaths, 249 million cases a year. So there's a strong justification on us looking at different platform that will enable us to eliminate malaria across the region. African Malaria Alliance last year gave a statement that we are facing a perfect storm where the tools that we're relying on are becoming very expensive. We have displaced population which cannot be addressed by the intervention we have. We have climate change that are actually making the problem much worse. But also the tools that we're relying on, people are not using it effectively, which is a question of compliance. And leave aside insecticide resistance and parasite resistance to the tools that we have. Gene drive has been in the space for a while. Over 50 years ago, people started speaking about this. But Lancet report in 2019, they projected by 2030 probably to be among the tools that we might rely on toward eliminating or eradicating malaria at a global scale. I think it was a good expectation or prediction. Probably we might go beyond 2030, but there is so much in the public domain in terms of development, which is quite encouraging on different groups continue working on this space. So what do we mean by gene drive? I guess all of us as human beings, we've been passing our genes to our next generation via what we call normal Mendelian inheritance. But gene drive is bringing a different concept where you can pass those traits or our genes quite faster within the population. So it's basically a preferential inheritance of your desired gene in a population nearly at 100%, rather than the 50% which is based on normal Mendelian inheritance. So what do we do? When you speak of gene drive, there's two ways you can do it. You can either target the germline cells, which is a sex-based cells, or you can target somatic cells, which are not sex-based cells. And different groups across the world, they are working on different formats. So people working on cancer space, they are looking at somatic cells where you make modification. That's address particular problem within a particular individual, but also you can target a germline where you make sure that whatever modification you do right now can likely be passed to the next generation at a very high frequency. There's different forms of gene drive, but these ones are the main ones where you have population separation, where basically you are, and Kayonda has touched on this, you're disrupting the genes that will basically crush the population down of your whatever target organism, in this case mosquitoes, or population modification, where you can modify mosquitoes and not to bite people, or in this case, we are trying to modify mosquito not to carry the malaria parasite, which is our biggest enemy anyway. So Transmission Zero, the program that we are running at Ifakara, is a collaborative global program, which is a collaboration between Imperial College London, Ifakara Health Institute, National Institute for Medical Research, and Swiss Tropical Public Health. It focuses on population modification and transgenic work from the inception to the entire test pipeline is happening at one of our branch, which is Bagamoyo. And we're planning to do a field site when time comes in the island of Ukerewe, located in Lake Victoria. We're already doing a lot of entomological studies in there. We're trying to look at population dynamics, population gene flows and how they disperse across different islands within Lake Victoria, and non-targeted organisms that are found in different breeding habitats exploited by mosquitoes. So how are we planning to test our technology? Basically, our technology is what we call separate split drive. We have two different forms. We have an effector molecule, which is responsible for blocking transmission within the mosquitoes. In this case, so you put it inside the mosquito, and the mosquito won't be able to catch the parasite. But you want to be able to push that modification from one mosquito to another mosquito, in this case, from one generation to another generation. And that's why you have what we call the drive. What we're expecting is to combine the effector strain that blocks the transmission with a drive strain, so you can be able to drive the effector, but also drive the drive itself. How does this happen? It's a very minimal modification. I think Professor Darlington mentioned you have different vectors of making modification, and it can be vector or non-vector. In this case, we are using what we call micro-injection technology, where you're injecting effector directly using a needle inside the egg of mosquitoes. So it's not using vector, and with that, we have a very minimum modification. And the minimum modification you have, the less likelihood you can develop resistance. There's a different stages of testing. So as I said, the effector will test if it can block the parasite. Then you'll have to combine the effector and the drive and see, and that's what will happen at the deployment. So you can be able to push it from one generation to another generation at high frequency. And these are different stages of testing our technology. We'll have phase one where we release genetically modified mosquitoes. These are mosquitoes that has only effector molecule which can block the malaria parasite development inside mosquitoes. And at this stage, there will be no gene drive. Basically, this will be inherited in a population via Mendelian inheritance. The expectation here is within a very short time, you're likely to lose your genetic modification because the inheritance will be at 50%. And then the likelihood of it spreading so far will be quite slow because there is no driving of that particular modification. And then we'll have what we call a middle stage where you have a static source of Cas9. In this case, you are driving the modification, but the driver is not driving itself. And at this stage, it's basically to test how can we push our effector beyond that release point. And then we'll have a final stage where you are driving the modification, but also the driver is driving itself. And that's where we have what we call self-sustaining. The expectation of this at this stage is that whatever you put in a population is likely to drive itself to a point where you can have a population which cannot kill the parasite at all. All this is nested within what has been proposed by WHO, that the testing of gene drive should be done it should be phase stages. Whether you start in the lab, which is the stage we are at the moment, then you go to the stem-free system, which is a containment large cage, then you do a small scale confined trial, which you go to the island, and at the end you are likely to do it in a 9-island space. We are championing 2 molecules that block the parasite. At the moment, we are just working with our candidate strain is what we call MCCP, is meganin melatonin carboxypeptidase strain. It's basically 2 compounds that have been extracted from African frog and European honeybees. And they've been engineered into the effector that we are putting inside mosquitoes. I'll skip this. So basically, this is a very short narrative. These are 2 strains that have effectors that I mentioned, meganin melatonin. And basically what we did, mosquito have its own immune system. What we have done, we have hijacked the mosquito immune system to express these 2 compounds. And what it happens is using messenger RNA as a central dogma, it tells the DNA what to do, and the protein will be produced. And those tiny compound will be inside the mosquitoes, and once mosquitoes are blood fed, the parasite development, which mosquito, that's to happen if once mosquito feed the blood, won't be able to happen because of these molecules that are already released in our body. So this shows that if you have the parasite inside mosquitoes and then you have this compound that has been released, those specific parasite inside the mosquito won't be able to develop. And this has been tested, that is true, it works. The mosquitoes that have been modified doesn't develop, their parasite doesn't develop compared to the non-ones that are not modified. And then, so what our future plan, once we be able to demonstrate that we can actually block this inside the lab, is started testing this at different stages in the field. And it depends on how successful we are at different stages of testing. Now, what I was explaining is we are at this stage where we have demonstrated that we have effector, it works inside the mosquitoes. If you are importing, you could have basically import this mosquito and test it in your local isolate by having parasite collected in the field and testing mosquitoes. And if this works, you continue to the field. And this infrastructure that we have in Bagamoyo is 2 containers that were imported. Initially, the idea of importing these containers from Europe was, once we do everything and it works in the lab, we can actually kill this container, take it to the field, instead of building another lab. But we have realized in the last 5 years, that's nearly impossible. They are there to stay, and if you're going to the field, you have to improvise or build a new containment facility. It was March last year where for the first time we demonstrated that we can actually create a transgenic mosquitoes in Tanzania. This is not a mosquito that blocks the parasite, it's quite late. So you need to create another 4 mosquito. You cross the 2, then you have the full product, which you have tested that it can block the parasite. But it was important for us to demonstrate that this can be done at ground level in Africa. Again, the next stage will be to test this in the semi-freeze system. This is another stage after from the lab you go to the semi-freeze, which is quite a little bit representative of what happened in the wild type. And this give you a room to control a population of mosquitoes rather than when you are doing it outside. So at the moment, this year in January, we had another 3 years grant where we are testing different things, basically looking for different effector molecules and drive. Beyond what we have, establish performance on safety profile for different effector molecules. As it is pointed in Cartagena protocol, there's a risk assessment for different products that we're working on gene drive. And we're expecting that in the next 3 years, we should have answers for some of these things. We already developed our stakeholder engagement and communication plan, which also fits within this current platform. At what time to communicate, who should we communicate with, and how often should we engage different stakeholders in the development pipeline. There is some challenges. Despite having all biosafety and national regulation perspective, you realize some of the things that are lacking is lack of context and technical awareness, understanding of GMO. And I think that's where it brings us together as different stakeholders. How best can we structure our communication and continue raising our awareness as a group of stakeholders rather than doing this in silo. And then you have insufficient framework to support assessment on the submitted dossier. When we started this work, it was submitted within competent authority and the Vice President Office in Tanzania, and we've been engaging with them in making sure that everything that are being done are done appropriately. And then there's leverage on existing ethical and governance guidelines on gene editing on ongoing trials. West Africa already have a regional platform on doing this, and East Africa, I think, is a space where we should be able to work to have, if you can have a regional governance guidelines to guide us toward working on genetic engineering. But also, we have different platforms that has been quite instrumental toward malaria control, and I think it's high time for us to start discussing this technology, even if they will be implemented 10 years, 7 years down the line. But I think we have an opportunity to start discussing them and realize what is required to be done now for us to be able to get there in 7, 10 years down the line. So this is all I had to share. Thank you very much.